All right, we're going to go over Chapter 14 on translation, and by the end of this chapter you should be able to describe the basic structure of proteins, explain the structure and function of tRNA, demonstrate the molecular mechanisms underlying translation, and then explain the one gene, one enzyme hypothesis. So we're going to break this up into 3 parts. The first part will be protein structure, the second part will be mechanisms of translation, and then finally the one gene, one protein hypothesis. So let's continue on with the central dogma. So we talked about DNA replication. DNA can make copies of DNA, but then the flow of information is that genes are transcribed into mRNAs, and then mRNAs are translated into polypeptides. So this is the part of the central dogma we're going to talk about now, translation, and then what exactly proteins are. So to understand a protein, you have to understand the basic building blocks of it, so amino acids. So amino acids are the subunits of a polypeptide chain, and so they are a kind of unique acid where they have 2 ends that can either be protonated or deprotonated. So this forms a chain, so a polypeptide chain can form. So the backbone of the protein chain goes in this direction, and each amino acid has a unique chemical moiety that is attached to the central carbon. So the R just is shorthand to indicate that it's variable depending on the amino acid, and there's 20 total amino acids that can form. So the nitrogen is where the polymer forms. So this is the nitrogen gets attached to the carbon. So in the event that you have a polypeptide chain form, you lose this OH, you lose one of these hydrogens, and a water molecule is produced. And then this nitrogen atom is covalently bonded to the carbon atom from the other subunit. So it creates a directionality. So before we talked about the directionality of DNA, the 5' to 3' end. And so when we talk about the directionality of a protein, we talk about the N terminus to the C terminus. So this is just kind of like the equivalent of the 5'. The N atom is what we refer to when we talk about the N terminus. And then there's a variable side chain here. So a peptide bond combines amino acids to form a polypeptide chain. So you can see the carbon right here. You can see the nitrogen right here. And then, as I said before, it's an acid. And so one part, it's kind of a unique acid where one part is positively charged, one part is negatively charged. And this gets covalently bonded, and the nitrogen bonds directly to the carbon. So you can see this double bonded O remains here. So the double bonded O remains here, but you lose this oxygen right here. And there's actually an H group attached as well, and then you lose the hydrogen here. So as an end result, you have an H2O group formed, water molecule formed, and now you've combined 2 amino acids together. So unlike DNA, which carries a negative charge, it's an acid that carries a negative charge because it's kind of this unique acid that has positive and negative sides. The end result is that the charge on a polypeptide chain from the backbone is negative, and the overall charge is determined by the side groups, the unique side groups. So depending on the levels of each amino acid that is used within a protein, you can have a net positive or negative charge. But it's not like DNA where it's negatively charged. We know DNA overall is negatively charged. Proteins are not the same way. So the directionality of the polypeptide chain follows the directionality of the mRNA. So if we look at the mRNA, there's a 5' to 3' end, and then translation occurs in this direction, going from 5' to 3'. So the AUG is the most 5' codon that is used, and then you go until you reach the stop codon at the 3' end. So we talked about the genetic code. We know that AUG, for example, encodes a methionine, and then we know how to take all of these codons and then translate them into amino acids that would be used, and these would all be covalently bonded together. So here we've seen the C to N bond kind of be used over and over in the polypeptide chain, and the directionality of the amino acids that are incorporated. Just remember that this is the N to C prime direction. So the N terminus is here, and then the C terminus is over here. So the N to C matches the 5 prime to 3 prime of the mRNA. And so you see we have this backbone that kind of moves through the entire polypeptide chain, and then each amino acid has a unique residue, R is for residue, that contains different chemical properties. So this is a valine right here, so it's made of a bunch of carbon and hydrogen molecules. So this is a hydrophobic type of residue, and that's because there's not a lot of opportunities to form hydrogen bonds on this. You can have rings. So tyrosine has this ring right here with a bunch of double bonds. Cysteine has a sulfur. So in previous chapters we talked about the cysteine bond that can form. So the cysteine bond forms when the sulfur molecules covalently bond together. So all of these different amino acids, you have this chain, but because they all have these unique residues, there's a lot of flexibility into the function of the protein. So there's 20 amino acids. They all have the carboxyl group and the amino group. That's the N and the C terminus part of the backbone. And then they all have an R radical group bound to a central carbon atom. And the R group of an amino acid determines the type of amino acid. So there's all sorts of ways that you can classify them. So there's nonpolar. These are hydrophobic. And so each of these side chains here on the alanine, valine, leucine, isoleucine, proline, methionine, phenylalanine, and tryptophan, these are all considered hydrophobic residues. And so what does that mean? These are residues that do not like to interact with water. And so when you have a protein, you have a chain that's kind of all wound up together. Some of it is on the inside, some of it is on the outside. And of course, all of this is surrounded by water molecules. So water molecules, if we go back to our chemistry, it's just simply an H2O. And so because the oxygen pulls the electrons from the hydrogen, you end up having a positive charge on the oxygen and a negative charge on the hydrogens. And so the water molecules want to interact because of that. So you'll notice that the alanine, so just using some of the basic chemistry of how it works, you notice that all of these atoms have a very similar electronegativity. And so these atoms do not end up having partial charges on them. So the C and the H's have the same electronegativity, and so there's not a lot of a negative charge on these. And so this is why they're hydrophobic, because if they're sticking into the water, they don't want to form any sort of bonds. And so instead of what happens, these tend to go towards the center of the protein where the water is excluded, or perhaps in transmembrane proteins, they go into the transmembrane where there aren't water molecules. The hydrophilic, these are the ones that like to interact with water, and so you notice that, for example, this has an oxygen on it, the threonine has an oxygen, the serine has an oxygen on it, and so these oxygen molecules will carry a partial charge, or the hydrogen will carry a partial charge, and so these residues can interact with the water molecules. And so these tend to be preferred to be not in transmembrane domains, and also they prefer to be on the exterior of the protein. You can also have polar, and so these might carry a positive charge or a negative charge, in the same way that a DNA, the phosphate carries a negative charge. So obviously if it carries a full positive charge or a full negative charge, then there's a lot of opportunity for it to interact with in hydrogen bond type ways, or some sort of interaction with other proteins or DNA. So you're certainly not expected to memorize all of these different residues, but you should be aware of the different principles that allows us to categorize them into these different ways. And you might notice different textbooks, different websites have slightly different categories for the different amino acids. So some of them might group all of these together, the tyrosine and the phenylalanine and the tryptophan together. because they all have these ring structures. And so these ring structures also provide certain chemistries in different situations. I'm just going to go by the textbook for this, but what I really want you to remember is the difference between things like hydrophobic, hydrophilic, and then those that carry the charges on them. So the primary structure, there's 4 levels of protein structure that we worry about for a protein. And these are called primary, secondary, tertiary, and quaternary. And so the primary structure is simply the sequence of amino acids. So we know that it's a polypeptide chain. And so this polypeptide chain can simply be written from the N-terminus. So this would be the N-terminus here. And then you would have a tryptophan and glutamic acid, an alanine, a cysteine, etc. So, you can simply write all of these amino acids in their sequence. And of course, this sequence is determined by the mRNA. Each one of these has 3 codons that were used to encode it. And so, you can go from the mRNA sequence to each of the predicted amino acids in the polypeptide chain. So the primary sequence is just simply the sequence of amino acids in order. The secondary sequence, now we're talking about the alpha helix and beta pleated sheets that are in the protein, and we'll talk in a second about what exactly those are. The tertiary structure is the exact 3-dimensional conformation. So these proteins do not exist as a single chain. They end up folding into very characteristic shapes. So we've seen lots of examples of these before. For example, the helicase, the row, these looked almost like barrels with a hole in the center. So that was because the tertiary shape of these molecules formed in such a way that they looked like a donut with a hole in the middle, for example. And each protein has its own unique tertiary state. So it's determined by the sequence. It's determined by the amino acid sequence. So this is just like the interactions between all of these different residues in the backbone determines the final structure, tertiary structure of the protein. But this is very hard to predict. So there's all sorts of different techniques that you can use to actually solve this. So crystal structures, solving crystal structures is a very powerful technique in biochemistry. And there's a lot of people that are interested in doing computational prediction of shape. And there's researchers at Georgia Tech as well. And then the final level of structure is quaternary structure. And so this happens when a protein, a macromolecule protein complex forms between multiple polypeptide chains. So we've seen this already with DNA polymerase, RNA polymerase, where it's constructed of multiple subunits, multiple peptide chains. And so when we talk about the quaternary structure, what we're talking about is how all of those individual units come together. So for secondary structure, we break it down into two primary types. And so this can either be an alpha helix or a beta pleated sheet, or just called a beta sheet. And so we see, why they're named. So this is an alpha helix. So, you've seen helices before in DNA. But DNA, just remember, is a double-stranded helix. There are two strands. The double helix, there are two molecules that are used to produce it. So an alpha helix is just a single-stranded helix. So it makes this characteristic helical shape, though, where it kind of goes around and around as it goes down in one direction. A beta pleated sheet, or a beta sheet, is just simply what occurs when you have a run of amino acids in a straight line, and then typically they bend back upon themselves in order to come back to interact with each other like this, and then come back and interact with each other like this. So this is secondary structure. So these alpha helices and these beta pleated sheets can be formed by a large number of different combinations of amino acids. So it's not like you have to have a specific set of amino acids that could produce the helix. There's lots of different combinations that can produce this helix. There's lots of combinations that can produce this beta pleated sheet, and we see this over and over again when we look in protein structures. And so, this is just an example of the polypeptide chain. So you notice that this is the chain, and so you should observe all of the N to C bonds, the nitrogen to carbon bonds, which is where the subunits come together. And so the R group is just shown right here. And so one thing you should notice for both alpha helix and beta pleated sheets is that they're held together by hydrogen bonds. So hydrogen bonds, again, continue to be important for the structure of DNA. It continues to be important for the structure of proteins. It's important for the interactions between proteins and DNA. And so the reason that these sheets form, or the helices form, is that there's hydrogen bonds formed together that stabilize these structures into these ways. But what you should notice is that the bonds themselves come from part of the backbone. So none of these hydrogen bonds are using the unique residues, the unique R groups that occur within each of the amino acids. So all of these side chains are available. They can bind to other things, but they're not actually being used in order to form the alpha helix. So, prediction of this structure in some ways depends on the R group, because the R group can inhibit the formation of this, but they're not actually used. They're not important for forming any of the bonds that hold these things together. So secondary structure, alpha helix and beta pleated sheet. Tertiary structure, now we're going beyond the alpha helix, and now we're looking at the whole three-dimensional form. And so this is an example of myoglobin. So myoglobin is important for binding oxygen, so it's used in your muscles, and so the muscles and cells will express this protein, and it can bind an oxygen molecule using a heme. So this heme is a cofactor. It's not actually part of the protein, but it's able to be bound to the protein, and it coordinates an iron that is useful for binding the oxygen. So these two red molecules are an O2 molecule. So the tertiary structure of this is kind of given by the exact shape of the protein itself. And so this is called a ribbon structure, and so the ribbon structure just shows the backbone of the protein, and you can see whenever it forms a helix, it looks almost like a helix, like you basically have something that's wrapping around like it looks like a helix. So this myoglobin has all sorts of alpha helixes. It doesn't have any sort of beta sheets within it, and you can break down the protein into where all of the alpha helixes are, but in order to truly understand the shape, you need to know exactly how all these different alpha helices are positioned within it. So you start with the sequence of myoglobin, then you identify all the alpha helices, and then you understand exactly how they're positioned in 3D space to get to the tertiary structure. Myoglobin is a single subunit, so it doesn't make sense to talk about quaternary structure for myoglobin, but if we look, for example, at hemoglobin, now we can also talk about quaternary structure. So myoglobin is used in order to bind oxygen in your muscle cells. Hemoglobin is what's used to transport it through your blood. So it's found in, red blood cells, and it is actually 4 different subunits. So all 4 of these subunits are able to coordinate a heme and a iron, and all 4 of these subunits are able to bind an oxygen. So each of these has like a little oxygen molecule that is bound to it as well. And so this hemoglobin is what's in your red blood cells, and this is what's transporting the oxygen from your lungs to your muscle and to your brain, etc. And so now when we talk about the tertiary structure, each one of these chains has its own tertiary structure, but we also have to know how these independent chains come together. So there's there's interactions between all four of the chains. We know that this part of the beta chain interacts with this part of the beta chain, and this part of the beta chain interacts with this part of the alpha chain. And so that description of how all of these different subunits are assembled together, that is what is referred to as the quaternary structure. So just make sure you know all four levels of the structure.